In this video, we will discuss the process of cell division and mitosis in a eukaryotic animal cell. In this first part, we can see a breakdown of the cell cycle. The cell cycle is broken down into two main phases, interphase and mitosis. Interphase is broken into G1, S, and G2. Mitosis is broken down into prophase, metaphase, anaphase, telophase, and cytokinesis. Interphase in action. The cell must first replicate its DNA so that both daughter cells can have identical copies to undergo mitosis. Chromatins then form to create sister chromatids, held together by centromeres. After interphase is complete and the cell has gone through all the checkpoints, it can then continue the cell cycle process and advance it to mitosis.
During prophase, the first step of mitosis, the spindle fibers and aster begin to form and the nuclear membrane begins to break apart, allowing the sister chromatids to move around in the cell. After the nuclear membrane breaks apart, metaphase begins and the pairs of sister chromatids line up in the middle of the cell along the equator. The spindle fibers then attach to each chromatid in the pair in order to set up for the next step of mitosis. After metaphase, the next step is anaphase, where the spindle fibers from each chromatid pull the pairs apart to the opposite sides of the cell. After the chromosomes have moved to the opposite side of the cell, telophase begins. Proteins in the center of the cell pull the membrane inward, forming a cleavage furrow and elongating the cell. During this phase, the nuclear membrane begins to reform around the newly split chromosomes. The last process of mitosis is cytokinesis, where the cytoplasm splits, forming two new identical daughter cells. Overall, mitosis is essential for cells in order to grow and develop and replace old non-functioning cells. Thank you for listening to our mitosis modeling video.